just warn myself, I suppose. So, all right. So it says it's recording. So once I get the file done, and then I'll email that to you for your records.
Thank you very much. Yeah.
No, thank you. And then so are we all accounted for? All my folks are here.
Okay.
Yeah, it should be 4 students from the Russian side, and I see, yeah,
3 from, So I got one voice, but yeah, I got Kane over there. Okay, we're good.
yeah,
He's gonna he gets to hear it in the room and over the internet. So, so anyways, just as a very brief introduction. So for today, we were just going to do group introductions and then talk briefly about our own research, as well as kind of the plan that Dennis and I have been talking about these last couple months, actually. What we're doing is, so Dennis and I actually knew each other maybe 8-9 years ago. We were roommates in Vancouver, Canada, when I was a graduate student. I think Dennis was already like maybe a recent graduate, and he was over there visiting, and he stayed with me for about 6 months, I believe. And so, so we I guess we stayed in contact in our research, more or less, it's similar field, I guess. And then I kept Dennis in mind, and we got this opportunity through HaRP, which is it's a human resources project that's located, I think its center, you can correct me if I'm wrong, Marina, but its central hub is in Hokkaido.
Hokkaido University, our university.
Yeah. And
the secretariat,
And then the idea of this is to promote collaboration and also business in different topics. So not only in industry that is strictly for business, but also in things like research in the environment. And so they're striving for a greater interaction, mixing things like research with business and economy. And I thought that our group perhaps could do something like this, at least over the next couple of years. So for today, I don't know if you guys have seen the outline. Let me go ahead and share what that looks like today. We have so this is the outline, the tentative schedule. And then Dennis and I will be leading this symposium. And what we're going to do is just a brief introduction from both Dennis and myself about our universities and our research. And then we just take a small break, and then also give the students time to introduce themselves to each other, because they'll be working together. And so that'll give everyone an opportunity to get to know the other side of this collaborative effort. And so let me go ahead and share screen again. Sorry, I'm having wait, where am I at here? It has oh, there we go. Okay. Sorry, I'm in Zoom mode here. Okay. So can you guys see my slides right here?
Yep.
The HaRP talk? Okay, perfect.
Yeah.
So let me first and foremost, I wanted to
Kevin, can I interrupt you for a second?
Yeah.
Just to double check that students, can you hear us? Dmitry, Artyom? Everything is okay? Nastya? Okay, yeah. It seems, yeah, everyone is here. So Kevin, please continue.
All right, yeah, thanks for sharing. So first and foremost, I wanted to say just at the beginning of this talk, just thank you to HaRP, and also a special thank you to Marina. Marina's joining us from the HaRP central office, the secretariat at Hokkaido University. She was actually the one that turned me on to this idea, which has led to hopefully many good things. So, and this project is it's supported with the idea of, I guess what I was saying earlier, creating business and research, in our particular case, opportunities between Japan and Russia. So again, thank you guys for doing that. And thank you, Marina, for helping us organize this symposium today.
Can I add a
yes.
few words?
Yes, go for it.
Sustainable development and environment-related research are a very important area of the HaRP supported efforts. And I think since a lot of, well, great emphasis is placed on it at Hokkaido University, we expect that its importance will grow. So what you are doing is, well, it falls into one of the priority categories. So thank you very much for coming up with the idea of holding this symposium. And we really look forward to progress in your joint efforts. And I personally will be happy to support you. Yeah.
All right.
Do what I can.
Thank you. Thank you very much. So, so just very briefly, in Japan, I'm sure you guys know exactly where Japan is. We're located on the North Island here in Hokkaido, specifically in Sapporo. Just for reference, down here is Tokyo and the islands of Okinawa, and actually Yonghaeng and Ranjani are located, I think, in Tokyo and Okinawa right now. So we got the different people from all over Japan doing their research. It's a little bit warmer, probably where they're at. It might be a little chilly, but right now in March, this is pretty much what Hokkaido looks like. And here's an interesting cultural exchange that in Japan, the snowmen actually only have 2 body parts, not 3, as in other parts. So I don't know how many body parts the Russian snowmen have. Is it 3 or 2? 3? Yeah. So in Japan, you see all these what I thought to be incomplete snowmen, but it turns out it's not that way. So, but we have very strong winters here too, and then so we're just getting over that into the spring. I've been working at Hokkaido University, and HaRP is central into or is centralized at Hokkaido University. It's the largest university by area in Japan. So it's right in the middle of Sapporo, and you can see this yellow outline of just how big the university is within the city center. It's quite gorgeous here, especially in the summer and fall times, and even in the winter if you're really like snow. A little bit about the university. It's was founded in 1876. It's one of Japan's top-ranking universities, and so within Japan, it's very nice to come here. They have it's a comprehensive university. So if you have students interested in the sciences or liberal arts at undergraduate and the graduate level, even master's and PhD, it's a good school to come to. So for international people who are interested in coming to Japan, Hokkaido University is or can be a really good option for that. So something to keep in mind. And typically we also say, instead of Hokkaido University, we shorten that to Hokudai. So it's something I slip into sometimes. And so when I say Hokudai, that's what I mean by that. So a little bit about Hokkaido. It has these pristine coastlines, and this is actually going to play into a little bit about what we're talking about today with the environment. But there's lots of things to do. They have summer beer gardens. They have this really famous winter snow festival that regretfully was canceled this year. But it's an international snow festival where they have multiple people come and build these really elaborate snow sculptures. And so you can see just the size of it with these people down here each year. It's really fantastic to go around and watch. So you want me, I'll go through the research part right now, and then Dennis can just go through his stuff as well after this. Okay, so I'll just, so my interest in, for research is in microbes. And I'm particularly interested in eukaryotes, and so especially the micro eukaryotes. And there you can see there's a whole variety of micro eukaryotes, and some of them have given rise to lineages that we're very familiar with, something like trees, maybe diatoms in the ocean, and even some parasitic groups that are very important ecologically for even us. And also, here's these white cliffs of Dover in England. Those are made of protist groups, so these coccolithophores. In the laboratory, I focus a lot on marine protostology. So this is just scraping together some sand and algae all over Japan, really. And so it's a lot of things to study out there. I do a lot of describing of biodiversity. So taking a lot of photographs and cataloging this diversity, trying to look at different morphological characters that make each species unique, and also doing some electron microscopy on it. One thing that I've been getting more and more into, this is a video. So this cell right here, this is a dinoflagellate. And then up here, there's some little algal cryptophyte. And this dinoflagellate is actually going to, oh, is it not? Oh, it didn't play it. Oh, no. Well, I guess the video became a little corrupted. Sorry, it didn't like it over Zoom, but it ends up drinking the cryptophyte, which is really cool looking. And then there's another organism that I was looking at that that had this harpoon structure on it. And maybe Dennis knows about this with Greg. We did this at UBC, where we were looking and we were trying to rebuild some of the evolutionary complex structures from these single cell eukaryotes. And we built these 3D models of these very complex nematocysts that were found in these dinoflagellates. And so that's one of my main interests. And the other interests, which we're going to be talking about today with the shellfish, are these parasites. So I studied the evolutionary history and diversity of apicomplexan parasites that are found in virtually all animals. My main question with these parasites is kind of what are they and where did they come from? So to understand how these parasites got into where they're at. So the alveolates in general are comprised of apicomplexa and dinoflagellates and ciliates. And this is just a very general view of that group as a whole. They're very diverse. And we say that every metazoan or every animal likely plays host to at least one apicomplexan parasite. And so this is kind of just a thought game that if we look at how many apicomplexans we have as humans, we have at least 4 different types. So we have Plasmodium and Babesia, which are these blood parasites like malaria. And we also have Toxoplasma, which is very important for, especially for women who become pregnant. If you have a kid, you go to the hospital, they'll give you a Toxoplasma test and tell you to stay away from cats and cat litter and don't touch stray cats because they don't want the babies to be infected with Toxoplasma, even though it's quite benign to anything other than a developing baby. And also there's Cryptosporidium. And so the reason we don't just drink water out of the river is that some of these apicomplexans can get into your gut and cause some problems for you as well. They're found in other organisms, so turtles, things like marine animals and seals. They're also found in coral, which I don't know if Dennis was working on that with Patrick at all a little bit. But then they're also very diverse in all these different types of invertebrates. And this is me and my PhD in Canada. This is Greg with his back turned to us there. I don't know if Dennis remembers him. And then this is the much thinner version of Kevin during the PhD, which Dennis probably remembers. And all the scuba diving there. And so what we did was we went around and I was looking for parasites infecting invertebrates. This is just a normal polychaete worm. And you can see within the gut right here where the arrows are, that these are these apicomplexan parasites that are found in the cross section of the worm. And they're usually found in this gut lumen area, but they're also in the coelomic spaces as well. And something in a dragonfly, you can see that in some of these organisms, these infections can be quite intense. And so not all infections from apicomplexans are so benign. A lot of them can be, in fact, very harmful. And this is very important for ecology and even for economics. Like we'll talk about the business aspect of things is parasites and shellfish, particularly those that are very important for aquaculture, can really influence the ability of businesses to make money. in that way. And so the way that we do this is some of the work we do with microscopes is we're isolating cells under a so we isolate the cells under a microscope, and we pick them up essentially one cell at a time in order to characterize them, to know what types of organisms that we're dealing with. And once you can actually manipulate the system, you can actually start to ask different questions. So you can start to ask, well, where did they come from, by using things like genomics. And you can also start to understand, what is their interaction with the host? So are they harmful? How do they speciate? So do they speciate within a host? Did they jump from one organism to another? And then we're also interested in finding very bizarre lineages as well. So things that may have an ancestral quality that lets us know where these parasites came from. And there's also quite bizarre organisms here. So these are also AP complexan parasites that, although they don't look like parasites, they are. And so this is the crux of the research, or at least the base of it. So for today, as part of this HaRP project, we were going to be focusing on shellfish. And these shellfish are these bivalves. And this work really started with this New Zealand group. This is here in Akechi, Japan. And we actually found a new AP complexan in shellfish. And this new APX is actually only found in New Zealand. And we went looking for it in other parts of the world because Japan and New Zealand a long time ago actually traded shellfish with each other. And we thought that the parasite might have actually started in Japan and then been transferred later to New Zealand. And so this work that we did was actually collecting different types of shellfish in the laboratory and just looking at it, trying to find these different parasites. And so this is actually pertained to a lot of field work, or it actually let us conduct a lot of field work all over in Oshoro at the different marine laboratories. And we are also able to make connections within the shellfish industry in Akechi and also in Lake Saroma. And we were able to get these lines of their oysters. And so that's something that I kind of hope to do also in this project as well, is to kind of reestablish that connection and get them involved into our research. What we ended up finding was that these parasites were localized to particular areas within the shellfish. Another thing we did was we wanted to actually find what is the life cycle of these organisms. So finding out how do they actually go from the environment into one host and into another, because many of these have two different hosts, and so they're able to spread. And the reason why we care so much about this is because heavy infections can actually cause shellfish to become sterile. And they can also cause the gametes, or at least their reproductive organs, not even to develop at all. So reproduction is essentially impossible. It's also impossible to eradicate. So once an infection of these AP complexans gets into shellfish, it's really impossible to get out, and the whole line or the whole brood has to essentially be thrown away. And so that's a huge economic loss for a lot of these shell fisheries. There's no known methods of prevention or control, and these things have been found in also a couple other species. And so there's also additive effects with other organisms. And so knowing how these parasites interact not only with the host, but also with other organisms is very important, both ecologically and economically. And so what I'd like to work on a little bit too with the hosts, and this is a bit more of an evolutionary question, is something about coevolution. And so this idea that every host has its own different parasite. And when we look at different areas of the world with these shellfish, to try to take a look at their morphologies and everything to see how they line up. So are these parasite, or the diversity of these parasites, is it a relic of host jumping events that have occurred almost randomly, or is it something where the host and the parasite have evolved congruently? And I think that's kind of important to understand the nature of the parasites as a whole in itself. So I'll let Dennis talk about his stuff, but anyways, yeah, thank you guys. Do you guys have any questions about the subject matter, or? No, everyone's good. Contented.
Yeah,
All right,
thanks,
so
thanks for very interesting
I'll let Dennis
presentation.
fire
Yeah.
away. Am I okay? I think I'm unshared.
Yeah. Do you have any idea how many
Oh,
lineages?
I think your microphone. Are you muted
Oh,
or?
really?
No, I
We
can
hear
hear
you fine.
Dennis.
Yeah.
Can you hear me now, Kevin?
Oh no, can't hear you.
So I'm.
Oh no, also no.
But you
I'm
can,
on.
Kevin, can you hear me? You can't hear anyone?
I can't hear Marina either. Let me see here.
Oh,
Did I don't think I did anything. I hope I didn't do anything. Can you guys hear me?
yeah.
Okay.
We can hear
Well, this is horrible.
everyone.
This is welcome to the future, guys.
Yeah.
Oh boy. Okay. 21st century problem right here. Oh boy.
Okay, I will type my question.
Oh no, I can't hear you. Oh no. Okay.
I will type.
What's going on? Oh, typing. Okay. Yeah.
Yeah.
We hear. Wait, is it just me? I'm the odd man out. This is horrible. Okay, I'm gonna change headpieces real quick. I really am sorry for that.
Yeah. Okay.
It turns out it was me. Okay, so I'm gonna change headpieces real quick.
Yeah.
Go ahead and start without me, okay?
The question, right?
Okay, that might have fixed the problem. Can somebody test it for me?
Yes,
Can you hear
can
me?
you hear
Okay,
me?
I can hear you now. Sorry, I think my computer just freaked out on me. Okay, sorry about that.
Yeah. I just wanted to ask, like, do you have any idea how many, like, yet undiscovered lineages of AP complex and parasites exists?
I would say that number is easily correlated with the number of species we don't have yet described as well. So I think right now there's probably about 2,000 species of AP complexans described. And if you think that the number of insects that are undescribed is in the tens of millions, there's probably tens of millions also of AP complexans just in insects that are undescribed. So the number of undescribed AP complexans is probably quite large.
Okay, yeah. And yeah, you said like these parasites are very important for shellfish industry, but
Yeah.
is do you know any methods of treatment from AP complex diseases for shellfish?
No, you know, it's a, in New Zealand and even in parts of Japan, when they get an infection, or even Europe rather, when they get an infection, they just pull the whole stock out of the water.
It's terrible.
Yeah, so they just waste it all and just start over, because it's cheaper to start over than to risk perpetuating the same mistake. So once they catch an error, or once they catch a disease, they'll just pull the whole line out and call it good. So it's just a loss for that year.
Okay, thanks.
Not a problem.
More questions from?
So you're saying about the intermediate hosts?
Yeah.
So usually there are around like 2 hosts, and currently, for example, in humans, you have like insect hosts for some AP complexes that is able to transfer to humans, right? So is there something similar going on for the marine one that you have seen in shellfish, like maybe fish or some other single cell eukaryotes?
Nobody really knows. It's such an underexplored area that where exactly they come from and how they get there, they can imagine it. They can probably imagine it's some type of other invertebrate that's obviously close by. But
because they are filter feeders, right? The oysters, so it could be free living and then like free living and then they go in become parasitic, because you did say it's in the lumen and stuff like that, right?
Yeah, well, AP complexins are obligate parasites.
Okay.
So that's one of they have yet to find an example of a free living AP complexin stage. However, they do have cyst stages that are in the environment. But it's not free living. It's not taking care of the cell. It's just it's kind of a survived capsule that gets ejected into the environment and then taken up by the host. And so it is, in a strict sense, yes, in the environment, but not as a free living organism that's moving on its own will.
I see. Okay, thank you.
No, good question. Thank you.
So thanks, Kevin. Yeah, so should I start my introduction?
Yeah, please. Let's do it.
Okay. I will try to share my screen right now. Can you see it?
Yes, perfect. Thank you.
So it's our pleasure to be here, our pleasure to see you despite of electricity shutdown and other issues. So yeah, let me introduce our research team. We are a group of protistology at the Institute for Biology of Inland Waters, so the Russian Academy of Science. We study mainly diversity, evolution, and ecology of protists. And our institute is located in the European part of Russia, in Yaroslavl region in Barok. This is a small settlement, about 2,000 people live where, and Barok situated on the bank of one of the largest reservoir on Volga river, which called Rybinsk Reservoir. And the settlement at our institute situated 300 kilometers away from Moscow to the north. Now I just show you some photos of Baroque and Institute. These photos were made by students. It's very beautiful place, situated within the trees, groups, and the name Baroque actually means small pine forest. It's a very good place for fishing, hunting, picking up mushrooms, and for any sort of relaxation, and of course for scientific investigation as well, in a peaceful place and in the quiet atmosphere. This is Baroque in winter, how it looks like right now. So our institute is one of the largest hydrobiological institutes in Russia. More than 300 people works here. There are 13 laboratories at the institute. Each laboratory study different groups of aquatic organisms, from small ones, from viruses, bacteria, phyto and zooplankton to fishes. And research topics of the institute include diversity of hydrobiomes, morphology, systematics, ecology, physiology, and biochemistry of aquatic organisms, structure and functioning of aquatic ecosystems, anthropogenic influence on water ecosystems, and invasion of non-indigenous species into water ecosystems. Our institute published Inland Water Biology journal with Springer, and you are welcome to submit your future papers to the journal. So our group deals with protists, with free-living protists mainly, which represent the greatest part of the eukaryotic diversity, and recent findings of the new species, high-level taxa, and even previously unknown big phylogenetic lineages of unicellular eukaryotes demonstrates that protists remain extremely poorly studied, but often represent ecologically very important ecologically and evolutionarily important organisms. And we are using microscopy and molecular methods to look at various groups of flagellates, heliazoans, testate amoebas, and ciliates. For example, we use various methods of scanning and transmission electron microscopy, as well as light microscopy to study the external morphology, structure of covering elements, and ultrastructure of cells of protists. And all these features, they have taxonomical significance, and the ultrastructure of protists is of great importance for elucidating the phylogenetic relationships between different lineages. Also, we sequence evolutionary conserved genes as well as transcriptomes and genomes of protists. We carry out phylogenetic investigations to reconstruct ancient evolutionary relationships. We investigate community structure in different types of water and soil ecosystems. We analyze species distribution in both global and micro scale in relation to various ecological factors, and trying to develop approaches to environmental monitoring and bioindication using protists. And all this assessment of protist diversity, features of their biology and ecology are extremely important for a deeper understanding of the role of these tiny organisms in the functioning and functional organization of natural ecosystems. So that's it for introduction, and now I would like to go to research. So the title of my presentation today, I would like to say a few words about evolutionary and ecological importance of free-living heterotrophic flagellates, the main object of our investigations. Heterotrophic flagellates are the least investigated component of the water ecosystems, but they play an important role in the ecosystem functioning. This is not a taxonomic group, but a polyphyletic assemblage of not related protists which possess flagellum or multiple flagella at least at one stage of the life cycle and characterized by heterotrophic feeding. Here you can see a picture of the eukaryotic relationships and diversity according the current understanding of eukaryotic tree, and please check these little drawings at the branches, and you can see that most of the branches on the tree are represented by flagellates. In general, all eukaryotes are subdivided into main domains, Amarthea and Diaphoreticus. So Amarthea includes uniflagellated apistacons, Breviatea and Apusomonadida. Altogether they called Obazoa and amoebozoans. Diaphoreticus, they are very diverse and include several supergroups of eukaryotes such as Archaeplastida, Cryptista, Gaptista, SAR. Famous group Excavate, currently considered is probably not monophyletic, and together with Scrums, Galactianids, Rigidiphylla and Mantamonas, they occupy some certain position on the tree of eukaryotes. And one of the biggest evolutionary problem which I'm going to discuss today is hidden within apistaconta. And this problem is the origin of multicellular animals. The origin of animals from their unicellular ancestors is one of the most important evolutionary transition in the history of life. The most developed model for the origin of animals or metazoans is based on a common ancestor with Hana flagellates. And current hypotheses say that multicellular animals evolved from the common ancestor with Hana flagellates more than 600 million years ago. At that, besides Hana flagellates, there are several more basal lineages of apistacons exists, like Phylasterians, Ichthyosporians, and Corallohydrium. They are very strange protists with specific lifestyle. Usually they are parasites or endocommensals, which make difficulties for construction of hypothesis of the origin of multicellular animals. We carried out a field work and discovered 3 new representatives of unicellular apistacons from Vietnam and Chile with similar morphology and lifestyle, but they are not closely related. So Pigaraptor vietnamica and Pigaraptor chileana belongs to Phylasteria, and Sesamonas multiformis form a clade with the mysterious organism Corallohydrium, and we call this new group Pluriformea. The discovered organisms have a variety of life forms, including flagellated stage, amoeba flagellates, and amoeboid cells. After feeding and hypertrophic growth, cell transformed to cyst. And palintomic division may be occurred inside the cyst, and up to 16 specimens arise as the result of division. Studied organisms are predators and attack cells of eukaryotic prey, and this process attracts many other individuals, probably by chemical signaling, and several predators can simultaneously, here is predators and he is a prey, so several predators can simultaneously feed on one cell of the prey, and as a result their neighboring cells can partially merge together. Solitary non-feeding cells also can form temporary cell aggregations, which are usually shapeless and easily disintegrate. And sometimes cells can merge completely and form multicellular syncytium-like structure. The budding of young flagellated daughter cells from syncytium was also observed. In all cases, the process of the formation of aggregations and cell fusion attracts other individuals in culture which try to join forming aggregation or cysts. Metazoan multicellularity is rooted in mechanisms of cell adhesion, signaling, and differentiation that evolved in the ancestors of metazoans. Transcriptomes of novel organisms contain homologs of metazone genes associated with multicellularity, such as fibronectins and laminins, as well as which is essential for cell and extracellular cell matrix engagement and signaling, as well as cadherins and C-types lectins, which are essential for cell-cell adhesion. Moreover, Pigaraptors contain genes of the embryonic development of animals, brachyury, which determines the formation of the central axis of the cell body from blastula, blastopore to horda. Recently, we isolated one more species of unicellular predatory flagellates, Tunicaraptor, which is not closely related to any known group of apistacons. The exact phylogenetic position of Tunicaraptor was not resolved even using multi-gene phylogenetic reconstructions. And Tunicaraptor possess similar morphology, lifestyle, and feeding mode as Pigaraptor and Cisamonas, and also contain genes of multicellular animals. So, for example, Tunicaraptor encodes homologs of the nervous system proteins, for example, repulsive guidance molecules, a protein which plays a critical role in the neurodevelopment and axon guidance in animals, which is used for treatment of neurological diseases, in particular Alzheimer's disease and multiple sclerosis. Also, Tunicaraptor has two main members of an ionotropic glutamate receptors. They are ligand-gated ion channels that mediate synaptic neurotransmission in animals. So discovered organisms form new phylogenetic lineages of apistacons and characterized by unusual and unique biology, morphology, and feeding mode for basal apistacons. They possess metazone-specific genes for cell adhesion, signaling, and development, which illustrate that genes of multicellularity arose before the emergence of multicellular animals, and novel organisms, unicellular organisms, have important behavioral precursors to multicellularity, like complex lifestyle, probable sexual process with fusion and budding of dotted cells and forming of aggregations. And interesting that feeding on large eukaryotic prey is an interesting and potentially powerful trigger in general for the formation and development of aggregates, for example, during joint feeding. And it's important also for clonal multicellularity due to hypertrophic growth followed by plentomic division, and perhaps this feeding playing a role in the origin of multicellularity in the ancestors of multicellular animals. Another big evolutionary problem related to investigation of heterotrophic flagellates is origin of photosynthesis and photosynthetic plants. So let's go to Archaeplastida, this supergroup of eukaryotes which unites red algae, Chloroplastida, which includes green algae and land plants, and glaucophytes algae. So altogether, archaeplastids have an enormous impact on terrestrial and aquatic ecosystems because they are main primary producers. But it is not clear how they evolved and how they uptake the ability to do photosynthesis. Their ancestors of all chloroplasts were cyanobacteria, This is prokaryotic photosynthetic organisms, and the history of plastids began with the fact that certain organisms absorbed cyanobacteria, which turned into chloroplasts. Such chloroplasts or plastids are called primary plastids, but it's not. But it's only a small part of the diversity of chloroplasts. There are multiple and independent enslavement of cells of red and green algae by our eukaryotes led to the emergence of organisms with secondary and tertiary plastids. Especially important in this and the symbiotic events involving the uptake of red algae resulted in the origin of such organisms as cryptophytes and gaptophytes and the various groups of heteroconto straminapile algae like brown algae, diatoms, xanthophytes, cynophytes, and also secondary plastid derived from red alga present in alveolates, for example, in dinoflagellates, chromerids, and parasitic epicomplexans. So the ancestor of archaeplastids was super important organisms for biosphere, but the early stage of this evolutionary transition, evolutionary pathway, they are very dark and not clear. Recently, we discovered the predatory flagellate Rhadelphis. One species was isolated from the marine littoral in Vietnam, and another one, freshwater, from a lake in Ukraine. Rhadelphis, let's see if I can play the video. Yes, so Rhadelphis look like a typical heterotrophic flagellate with two flagella. Their cells are about 10 micrometers long. They are slightly flattened and rigid, and Rhadelphis is a predator and actively feeds on other flagellates such as marine and freshwater botanids. However, phylogenetic analysis surprisingly showed that Rhadelphis form a sister lineage to Rhodophyta, to red algae. In other words, Rhadelphis is a part of Archaeplastida supergroup and located rather close to its common ancestor on the tree. Meanwhile, Rhadelphis, as you see, doesn't look like a plant or alga. It's active predator feeding this phagocytose and capture eukaryotes and bacteria. And this may indicate that the ancestors of plants were predators with phagocytosis. But what about the plastids? All archaeplastids have them, and they were inherited from their common ancestor. This means that Rhadelphis should have plastids too, but electron microscopy didn't reveal plastids in Rhadelphis. However, some plastid genes were found in the transcriptomes of Rhadelphis. These are, for example, genes of TIC-TOC complexes, as well as ferredoxin and ferredoxin reductase, which are involved in the assembly of iron sulfide clusters and heme biosynthesis. So this indicates that Rhadelphis has a rudimentary, not photosynthetic plastid. To summarize, Rhadelphis can be characterized as a unicellular predatory plant that clarifies the sequence of ancient evolution. And obviously it started from the predatory flagellates, which simply captured cyanobacteria as a food object. Then cyanobacteria became a symbiont at the stage of mixotrophic feeding began. We also have to conclude that the common ancestors of archaeplastids could be only mixotrophic. Later on, mixotrophy disappeared, and today we know only photosynthetic algae and plants that feed by photosynthesis and heterotrophic radelphis. And the ancestor of Archaeplastida may have also resembled radelphis cell morphologically, because it's easy to imagine that predatory flagellate like radelphis capable to feed on large 6 to 9 micrometers prey could engulf a large cyanobacteria, which became a primary plastid. It was very funny to see that after the publication on this predatory process, some journalists come up with funny idea that ancient mother of algae and radelphis resembled a tiny version of a human eating tree feed, the mobile carnivorous plant from the novel of John Wyndham, The Day of the Tree Feeds. And the last big evolution equation which I am going to discuss today is origin of parasitism. Let's go to alveolates and illustrate this problem. Most alveolates fall into one of three main well-known subgroups, which is phagotrophic ciliates, predominantly photosynthetic dinoflagellates, and parasitic apicomplexans. Alveolate evolution and origin of these groups has been intertwined with a question of the broad general interest, the origin of parasitism. Recently, it was shown that closest relatives of dangerous epicomplexans parasites, which Kevin just told, and they causing malaria, for example. So the closest relative of these dangerous parasites are peaceful photosynthetic algae, chromerids. And it was a big surprise and a big discovery. You can see it was published in Nature. At the same time, we investigated colpodelids. These are heterotrophic flagellates which suck out the contents of their eukaryotic prey, and this type of nutrition called mesocytosis, and this type of nutrition is used also by apicomplexans. So colpodelids have a microtubular canoid associated with rhoptria and micronemes, and these organelles are similar to the organelles of the apical complex of parasitic apicomplexans. Our phylogenetic reconstructions show that predatory flagellates and chromerids form a monophyletic group, which we call chrompodelids, and this group is sister to apicomplexan parasites. It's interesting that colpodelids, they are heterotrophic, but they contain plastid targeted genes in the transcriptomes. Also, both chromerids and colpodelids, they possess many protein coding genes which were previously considered to be specific for epicomplexan parasites. For example, proteins associated with the division of parasites or the oocyst membrane, as well as associated with organelles of the apical complex. This is, for example, proteins of rhoptria and micronemes, and cytoskeletal SAS6L, and these genes determining the possibility of a parasitic lifestyle of the AP complexons, and they are already present in photosynthetic relatives of these organisms. So, thus, an important condition for the origin of AP-complexans parasites was the presence of pre-adaptive features of the cells of in the cells of their ancestors, in particular the microtubular ganoid and dropteria, which were initially associated with heterotrophic feeding and predation on eukaryotic prey. But what about the common ancestor of alveolates? We have studied colponimids, which attacks smaller flagellates and capturing them entirely by their longitudinal feeding groove with undulating posterior flagellum, and multi-gene phylogeny based on more than 300 genes undoubtedly indicates the position of colponimids as a basal lineage to all other alveolates. Taking into account phylogenetic placement of colponimids, it's possible to suppose that the ancestor of alveolates was a free-living predator containing two heterodynamic flagella, a posterior digestive vacuole, and extrusive organelles for active prey capturing, situated between the submembrane alveolar vesicles. From the ecological perspective, heterotrophic flagellates are the major part of the microbial food webs. They consume bacteria, dissolved organic matter, detritus. They feed on cells of our small protists. At the same time, heterotrophic flagellates serve as a food source for ciliates and micrometazoans. So heterotrophic flagellates should be studied more precisely for better understanding of structure and functioning of water ecosystems. But what do we know about their real diversity in water? And the answer is not much. And the good example came from the Tara Ocean expedition. The aim of this expedition was to collect samples around the globe and sequence DNA, environmental DNA from samples from sea plankton. So the expedition took 3.5 years, and samples were taken from 210 sites from the world ocean. The data of the analysis of environmental DNA sequence showed that the real biological diversity of the protists can be up to 9 times higher than our current estimation. So we know only currently know only 1/10 of the real diversity of unicellular organisms, and this is a huge field for future research, which will contribute to our understanding of structure and functioning of ecosystems. Thank you for your attention.
Oh, so anybody have any questions for Dennis? I actually have a question. You showed I guess this phylogenomics. There's always a new tree that comes out every, with a new supergroup, like this crumbs group. That's the first time I've heard of it. That must be relatively new, I suppose. But how complete or how many more changes do you feel that current phylogenomics could make to this type of tree? Like, what do you think needs to be done to actually make it resolve?
So it's like two opposite processes. For example, new lineages of eukaryotes, they are constantly discovered, and every year something appears again. Oh, like new lineage of eukaryotes, new supergroup. But at the same time, with the development of methods of phylogenomic and the with the increasing of genomic and transcriptomic data, we know more and more about the sister relationships of the groups.
Okay.
And so with in future, I think many of these super groups will be collapsed together, and yes, like.
So you see, I think yeah, as time goes on and more technology is made better, it'll resolve itself. Yeah, okay.
Yeah, of course. Like we constantly discover like new lineages, but with the development of modern tools, we will find the evolutionary place on the current tree, and we'll join some already established supergroups, I guess.
Yeah, that's really neat. Anybody else have any questions for Dennis?
Can I ask a question?
Oh yeah, please.
Yeah.
Although, as you can perhaps guess, it will not be related to the substance of your research. I learned a lot of new things, but definitely not sufficient to be able to ask, targeted question. My question is more related to the educational side of your project. So what do you think you could do in Japan? It's a new academic year to come, as you know, 2021, 2022, ending in March 2022. So from these two presentations of Kevin's and your, Dennis, so what do you think? How can further research, your further field work in this area contribute to the research work of students present here today and probably joining you in the future?
Okay.
So yeah,
I
just wanted to hear both sides if possible.
Yeah, I'll go first, Dennis, and then you can follow up. So directly, at least in the laboratory right now, Yonghang and Keihen and Ranjani are doing their fourth year projects and their master's projects respectively. And their projects actually, directly relate to the shellfish bivalves in the case of Ranjani, and also indirectly as far as other protist groups and bioinformatics and sequencing with Yonghang and Keihen. And so at least one student would probably be getting their master's in this topic of research. And then the other, so Keihen and Yonghang are going to be doing also probably collaborating on these publications. And so the idea would be that they would have not only their thesis, but also these international publications that we can publish together. And that's really means a lot as far as even if you're not into science or want to go further in your career, having these academic publications is kind of like evidence of that the student did something productive and was able to do something meaningful that was accepted by the community. So those are the two things. Furthermore, so for the education, well, the coronavirus thing is always really, that makes scheduling things hard, but I was hopeful that in the maybe even next year or into the future that Dennis would also participate in the Hokkaido Summer Institute, where we bring researchers from abroad to Japan for a week or 2 weeks, and we conduct a workshop in the laboratory, and that's something that we can share. And there's also, I guess in the longer thing, there's JSPS and Russian Academy of Sciences. There's bilateral grants that exist. And so there's ways to sustain collaboration, but also the student education. You want to add anything, Dennis?
Yeah, like I do this presentation in the universities and, for example, show this current tree of eukaryotes, and it looks weird for students, but it's very interesting because everybody remember kingdoms of animals, kingdom plants, kingdom fungi, and kingdom protists. So they not exist anymore. Like it's
Oh my
replaced
God.
by this
Wow.
bush, this super complex tree, but supplied with videos, is nice looking videos of protists. It attracts attention, and more students are interested in investigation of protists, and they are coming to the lab, especially they can do like summer training courses, and they teach them microscopy methods, and they can observe protists, and this is a source for attraction of students to this field and to academician institutes. Yeah.
We hope that our very humble contribution to your joint project, that the purchase of the PCR machine and that microscope can hopefully in some small way lead to further progress. Do you think you can, I have no idea what the Russian or Japanese policy with respect to, for example, mailing samples. So do you think you can exchange, or will you exchange data? I'm sorry, I'm very ignorant in your area.
That's also, so in the past, my experience actually with getting samples from Russia, it can be quite tricky having biological samples actually mailed from Russia to Japan, or even into Russia from Japan. I don't know, Dennis probably knows a lot more of that, since you probably
it's very tricky, like
and so usually this is why I think this is why Dennis goes directly himself to Vancouver, I'd imagine, with the samples and the cultures.
It's
because
not possible to ship anything using express post. It's possible to do like Russian General Post. They are not so strict, but you know how time consuming is this.
How many months? Sometimes it just gets lost.
Yeah.
From my experience. Do you
It will
think
take 1 month at least.
So you'll be exchanging data mostly, right?
Yeah. And so for
based
time,
on the on the field studies.
Yeah, and so based on the field studies, until traveling is easier between the two countries, then perhaps we could have more of a kind of in-person collaboration.
Yeah, and it's not a problem to take samples in Russia and take it to Japan, and it's not a problem to take sample in Japan in person and bring it to the plane and go back to Russia. This is okay, but
it's crazy. Sorry.
Yeah, we do a lot of
very logical.
Yeah, but it's whenever you go through the protist people's backpacks and suitcases, inevitably you find samples. It's we're kind of world class smugglers of samples in tubes.
Okay, so
this is still recording too, so everyone knows the secret.
Kevin told me that the industry people were interested in further in some involvement in this project, because as you mentioned at the very beginning in your presentation, Kevin, they had problem. They basically had to obliterate the whole populations of their culture if it gets infected, right? Did I get it correctly?
Oh, and
so for them it's a very urgent issue, right? If they,
that's often the case, and I haven't had that. I know it probably happens in Japan, but the in New Zealand and in Europe, that's the common method. That if there's a problem, they just pull the entire stock and I'm sure it's probably the same for Japan.
Well, they do it with chicken, so very likely that they would do it with shellfish.
Yeah, because it's so hard to, usually these things like, to get rid of certain types of cyanobacteria and stuff, there's like special genes or antibiotics you can use, but in things like these protists, these eukaryotes, it's very hard to kill them without harming the eukaryotic host as well. That's one downside, is the phylogenetic proximity of the host and the parasite. It's very close, so it's hard to get a difference, really, even though they look quite different. And then I guess the other part of it is environmentally, who wants to dump thousands of liters of antibiotics into the ocean, right? That's another harm. So as aquaculture practices become larger, these issues become a little more complex.
Well, which is promising in terms of further industry involvement, right? And
yeah, I mean,
probably financial aid, additional funding from the interested companies as well, right?
Yeah, I mean,
Well,
there's always no, that was the thing with the you know, if you know more about the parasite, you know exactly what causes the harm or what's killing it, because correlation is not always causation. So oftentimes when you see something dying, you will see the bacteria on it, but it may not be the bacteria that's killing it. It might be something that caused it. So having a better understanding of what's causing these things, and not just what we obviously can see, is actually very helpful in that regard. So anybody have any other questions for Dennis or education or anything else for?
Are we going to continue?
Ranjani.
Basically the same.
Yeah, Ranjani has a question.
I had a question about the expedition. Like in all the sites that you collected samples from, I noticed that there weren't any near New Zealand or like Australia or Japan. I was wondering if there was any reason for that.
Is it Dennis or?
I think to
Dennis.
you.
For me.
Oh no, for Dennis.
Dennis. Yeah. Like all my stars are near Japan.
Yeah, of course, like for us, it would make sense to collect samples everywhere, because like natural ecosystems are full of new processes, and sometimes these new processes can be sister lineages of some important organisms. For example, sister lineage to some important parasites, and understanding the biology and genomic organization of the living organisms is very useful for understanding the biology and mechanisms of pathogenicity of parasites. So yes, sampling is very essential, especially in the remote areas, so which were not studied before.
Thank you.
So I think we're a little bit past 5 o'clock, and so we had up on the schedule, we had some student presentations. I know Yohei, Ranjani, and Kehen had prepared some talks. And let me go back. Sorry, I didn't have the. I had a million windows on my browser open. I'm very sorry. Oh, there we go. So I think Ranjani, are you prepared to give your presentation?
Yes.
All right.
So is it okay if I share my screen?
Yeah. Okay. Is that possible? Or?
Yes. It says the host has disabled screen sharing.
Oh boy. Okay. How does this?
You can enable that, would be great.
Oh no. Screen share. Oh, there we go. Okay.
Thank you. Oh, it still doesn't allow me.
Oh no. Oh wait. Is sharing only? Okay. Try that. Does that go?
Okay, it works.
Okay.
Okay. So hi everyone, I'm Ranjani and I'm from India. I am currently a 4th-year undergraduate student, but from April, I'll be a graduate student at the Graduate School of Science in Hokkaido University. And for my undergraduate research, I did my project on songbirds, so basically neurobiology. And I looked at the differential expression of genes in 2 kinds of songbirds called closed and open-ended learner songbirds. And I saw if there were any differences. And from April, I'll be studying apicomplexins in shellfish. And we're first going to sample 4 kinds of shellfish. So they're the mussels, surf clam, geoduck, and scallops from different parts of Japan, like Otaro and Hakodate, and all around Japan. So like Hiroshima, Enoshima, and even Okinawa. And then we're going to collect them and dissect them and then fix them in wax, and then cut them into thin slices, and then use something called LCM, or laser capture microscope, to cut the apicomplexins from the tissue. And then we're going to do sequencing of the 18S rDNA to know what exactly, what kind of apicomplexes we're dealing with. So this is a small video about how the LCM works. So as you can see, this is the LCM, and now you can use laser to cut specific parts of the tissue section. And using this, we can isolate areas which contain apicomplexins, and then we can amplify their DNA and then analyze them. Oops, it's playing again, sorry. Oops, okay. So this is how the LCM works. And I'm looking forward to collaborate with everyone in this. Okay, thank you.
Thank you very much. Anybody have any questions for Ranjani?
Ranjani, do you have this laser section machine at Hokkaido University?
Yes, we have a common one for the entire like graduate school, and I think we can use it. There's not many people using it.
And am I understand this right, that you can make like a section, visualize parasite, and somehow pick this cell for single cell, genomic, transcriptomic?
I don't think we're doing single cell, but we can definitely collect the apicomplexin from the section.
So it's some like technique which can take the parasite off, right?
Yeah,
Mm-hmm.
yeah. So it's actually, it uses a light, like was it infrared laser or UV? Which one pushes it? Which one picks it up? I forget. But it's infrared and UV. Yeah, it's infrared and UV. So one of them cuts it, the sample, and the other one pushes the lid down and like sticks to it and picks it up again. And so you can cut. They used it or developed it for cancer cells and things like that. That's where I think got a start. And so you just take a wax section, and from those wax sections, you can find something under a microscope and then go to its sister section and then do the laser dissection on that sister section in order to genotype it. Or even for, like you were saying, transcriptomic or other, because Ranjani, you've used it before, but did like genome analysis?
I did that to like get certain brain areas. I was looking to get certain like very specific cells from that brain area. So
yeah, and you
different, but I think quite similar still.
Yeah, and you did the RNA stuff, or was it the genomic?
I didn't get to that part because initially I thought that I would continue in that graduate school, but then my plan changed, so I didn't really continue with that. But I've done it once.
Okay. But yeah, you can do a number. It depends on how the cells are fixed too. So if you fix it, something that preserves like the DNA. So instead of formalin fixation, use something like methanol, and that would preserve DNA a little bit better.
Okay, thanks.
All right, so thank you, Ranjani, very much. That was a great presentation. Next, Yonghang is from Okinawa. Please say you're able to share your slides.
Yeah, I'm able to.
I've shown some pretty bad incompetence to my technology.
You guys can see my screen, right?
Yes, sir. Thank you.
Okay, so yeah, I'm from Singapore. I'm currently studying in Hokkaido University. I'll be in my 4th year this coming April. The picture on the right here you see is actually Singapore. And I'm usually in Hokkaido, which is very beautiful. So occasionally I go into the mountains just to see a view from higher up. Currently I am in Okinawa to do my internship. So because Okinawa doesn't really have many mountains, I sit on top of a rock on the beach, but yeah. So what I've been doing so far is in Hokkaido, I've been doing taking a look at the diversity and the toxicity of benthic dinoflagellate Kulia. So these guys are found in quite a lot of places, like subtropical, tropical areas, and I've been looking at their diversity in terms of at what locations, what are present. In addition to that, I'm also taking a look at their toxicity, because these guys actually do produce Kulia toxin. So you can see the interesting thing is within a species, they have a different expression of the toxins. Like for example, we have one which is a specimen from Guam. It's actually not producing any toxin. However, Kulia meliensis, this group is usually known to be toxic, as well as another one that we found, another phylo group of Kulia canariensis. This group is usually not toxic, but we found another group that is actually producing toxins in Guam itself. So we are not sure exactly whether it is based on location or based on what environment it is grown in. But so this is one of the projects that I'm doing now. In Okinawa itself, I am actually working on the endosymbionts of another dinoflagellate, which is Synophysis. So these guys actually have cyanobacteria endosymbionts as well as other bacterial endosymbionts. So I'm here trying to get the information on whatever genes they have, nuclear or whatever genes the cyanobacteria has, to see at what stage of endosymbionts, at what stage of the symbiosis it's in, whether it is obligate or whether it is being consumed by the dinoflagellate itself. Another project that I'm doing here is actually Ostreopsis, which is another dinoflagellate. You guys will see a trend that I'm doing a lot of dinoflagellates. But so I'm looking at the Ostreopsis, which is a common, one of the dinoflagellates that is commonly known to produce toxins as well. And these toxins or these dinoflagellates that produce toxins has also known to have associated bacteria with them, which can influence the production of toxicity. So I have been screening the samples collected from around Okinawa, and I will be screening their toxin production as well as trying to identify their bacterial epibionts. And in the future, hopefully, if I have enough time and able to, and if they grow well, I want to actually reintroduce like different groups of bacteria and see if and how that actually affects their toxin production. Yeah, so thank you.
Thank you very much. Any questions?
If you guys have any questions.
Sure.
Thanks. I want to ask about Kulia.
Yes.
Yeah, very cool name for dinoflagellate. And yeah, you said like it's toxic. Can this dinoflagellate be a reason of toxic tides, like red tides?
So Kulia has been associated with other dinoflagellates that cause the red tides, but I don't think it, at least not from what I've read, that it's not the main one of the main sources of the toxicity. So usually it is with other dinoflagellates that you see the harmful algal blooms actually affecting like humans and marine life. But it is definitely producing certain toxins, because when there are bioassays that have shown to actually been that the toxins have actually shown to actually kill off certain cells like artemia, as well as some I think that they have done mouse bioassays as well.
Thanks. And do you know something about the main environmental factors which stimulate blooms of toxic dinoflagellates and causing red tides?
So from what I know, it is that is the runoff, like for example, eutrophication, if I'm pronouncing that right,
Yeah.
that can influence the dinoflagellate blooms, as well as temperature has also been shown to play a role. But I think that's more or less what I know.
Thanks.
No problem.
Yeah, thank you very much, Johan. All right, and then I think next up is Kahan. I was gonna.
Does my voice feedback into the other mic?
Oh, sorry, I shut off the.
All right. I'll share my screen right now. Give me a moment. Okay, can you guys see?
Yep.
All right. Okay, so my name is Kahan. I'm from Singapore, same as Yong Heng, and I'm gonna start my 4th year in April. So first things first, let me just talk about what I'll be doing in my 4th year, which is similar to Yong Heng. I'm going to investigate the different toxic compounds produced by the dinoflagellates in the algal blooms, specifically Ostropsis. So just some background information for you guys. So what's so important about these algal blooms? Because they produce toxins that are very harmful not only to our ecology and also to our economy. So what I mean by this is that these toxins produced by the dinoflagellates, they can cause they are a health hazard when they enter your body, and they do so primarily by 2 ways. First is by consuming, for humans, consuming the animals that have a lot of these toxins built up in their liver, for example, or by breathing in aerosols that contain these toxins. So, and also if we know what kind of what are the compounds responsible for the toxicity, we are able to identify these compounds in shellfish and sort of prevent these poisoning events from happening. So previous research done by our laboratory identified I think five different types of clear toxins in just one species, which highlights the diversity of the type of toxins produced by the dinoflagellates. So even within one species, you see such a large diversity. Yeah, and using the high pressure liquid chromatography, we're able to identify even more chemical compounds within just one species of dinoflagellates, which leads us to believe that in the species that are in the genus that I'll be working on, Ostropsis, there will be many types of many different analogs of the toxins that will be present. Yeah, so one of the objectives of my project is to identify what exactly is the toxin, and also through transcriptome analysis identify in what ways it is being produced, how it is being produced. Is it being produced by the dinoflagellate itself, or is it produced by the dinoflagellate and then passed on to the bacteria to be processed, and then it turns into toxin? Yeah. So also, as what Yong Heng mentioned in his slides just now, Ostropsis are known to have bacterial symbionts associated with it. So another sort of like a side goal of it is to identify exactly where the bacteria are located. Is it located inside the cell, or is it located outside the cell? And we plan to sort of elucidate this by electron microscopy. Yeah. So yeah, that's it for me. Any questions for you guys?
Thanks. Which method did you use to determine chemical compound of Coolea?
Oh, we use the high performance or high pressure liquid chromatography. So we first we extract the toxins from the sample from the culture, and then after that we extract like the raw extract from the culture, and then we put it into the machine to sort of process it in these which gives you a graph like this. So you can see like the different like molecular masses of the different compounds within that extract.
Okay, great. Thanks.
Yeah, so any other questions?
Any other questions?
My voice.
You're muted.
We can't hear you, Kevin. Keegan muted himself.
Sorry, Kahan and I have to do a microphone dance because we're in the same we have a room right here and he's on the other side of it. So my voice will pick up on his microphone. So anyways, Kahan, I was gonna ask you, what type of, given the diversity, or because Kahan actually just started working here 3 weeks ago. So it's actually, he's just beginning. And so in your expectation, how, I guess, how diverse, just in the first 3 weeks that you've been here, is there just more diversity of these toxins and dinoflagellates that you imagined, or what was your expectation? And just 3 weeks in, what have you found out? What's your biggest surprise, I guess?
Well, my voice, is it okay? Okay. So, yeah, I initially thought that, one species or even just within that whole genus itself, it produces like just one toxin. That was my preconception at the start. But after reading through the different papers and, yeah, basically the other results that has been published, yeah, I was quite surprised to know that even one species, there are like so many different types of I guess toxic compounds that can be produced. Although we are not really sure if the compounds itself are toxic, we have to do some bioassays for that. But currently we are able to identify many analogous compounds. So yeah, that was something that I wasn't expecting. And for my project, I would also expect to see something similar. Yeah.
Oh, thank you very much. So, does anybody else have any? Oh,
Yeah.
sorry.
So just sorry. One thing I'd like to add is the one thing that I found recently is especially for the Coolea maliensis, the one that we have, the toxin production also, I think, changes within the same strain, can change depending on culture conditions, so that the amount or the variety of toxin can also change. So I think that's also something we should take note of.
Yeah, definitely.
All right, great idea. Thank you guys for your questions. So now I think Dennis is the first one up on the Anastasia. Am I saying your name correctly?
Yes. Hi.
I'm probably butchering that. Anyways, very nice to meet you. So you have a presentation for us as well?
Yeah, it will be four presentations, and I have to admit that for all students, it will be first time they present their results in English.
Okay.
So,
There's no
Good luck.
shame in that. Good luck. Great,
Yeah.
great advisor. All right, bye. Here's your parachute. All right. Anyways, thank you. Please.
Is everything okay? Can you see my screen? Yes. Okay.
Perfect.
So I'm Nastya. I live in Baroque and I work as a lab assistant in a laboratory of microbiology. And I'm also getting my master's degree at Voronezh State University. And today I want to tell you about some interesting flagellates which call telonemia and probably play a big role in eukaryotic evolution and microbial communities. The broad scale tree of eukaryotes is constantly improving, but the evolutionary origin of several major groups remains unknown. Resolving the phylogenetic position of these orphan groups is important, especially those that originated early in evolution, because they represent missing evolutionary links between established groups. And telonemia is one such orphan taxon for which little is known. Recently, based on the transcriptomic sequencing and phylogenomic constructions, it was established that telonemia is a sister lineage to SAR. SAR is the largest supergroup of eukaryotes, including heterocont algae, for example, diatoms and brown algae, dinoflagellates, ciliates, apicomplexans, foraminifera, and radiolarians, and many other organisms. Based on the topology of the tree, a new supergroup SAR was proposed. There was no morphological evidence of the SAR ancestor, since no modern phylogenetic lineage sister to this supergroup were known. And now we know that telonemia are close relative to SAR. We have researched some flagellated telonemids, and most species of telonemids are protists with two sub-equal flagella inserting at the base of the cone-shaped anterior end, which forms rostrum. Probably this morphological structure is ancestral in relation to taxa within SAR. And we also know that telonemids feed on other protists and bacteria, and that microbial eukaryotes form the basis of their diet. Despite the low abundance, these protists can be a significant part of the structure of microbial food webs. To date, several hundreds of environmental sequences belonging to telonemia were known, but only 2 species have been known and described, morphologically described. This is Telonemma subtili and Lateranema antarctica from sea waters. We have isolated 6 new clones of telonemids. These are clones pipe 1, pipe 2, and TL1 from the Kara Sea, Russia. Freshwater clone TL2 from Champ, Island, France, Joseph Land, Russia. Sea clone TL3 from White Sea, Russia, and fresh clone TL4 from Sakha River, Adygea, Russia. And we have investigated the external morphology of the discovered protists using phase contrast, differential interference contrast, microscopy, and scanning electron microscopy. Based on the environmental sequence data, we have researched the diversity and phylogeny of telonemia. And according to the results of the morphological analysis and topology of the 18S rRNA tree, we're supposed to describe the TL2, 3, and 4 as new species of the Telonema genus and to isolate pipe 1 and 2 in the new genus of Telonema with 2 new species. Furthermore, the influence of temperature and salinity on growth dynamics has been experimentally ascertained for some clans. For the pipe 1 clan, it was found that the optimal temperature for cell growth is 10 degrees. The optimum salinity for the pipe 1 clone is 20 per million. In both fresh and hyperhelium medium, cells die already on the second day of the experiment. The optimum temperature for cultivating cells of the TL1 clone is 10 degrees. And in the experiment to identify the optimal salinity of the medium for the TL1 clone, the maximum number of cells was recorded at 30 per million on the fourth day of the experiment. At salinities of 0, 5, 10, 50, and 100 per milli, a decrease in the number of both clones was detected already on the second day of the experiment. Thus, the results of our research have shown that Telonema prefer cold marine ecosystems, which is confirmed by data on environmental sequences from various geographic areas. For a long time, it was considered that telonemes are exclusively marine organisms. But with the development of NGC methods, a number of environmental freshwater sequences closely related to telonemes have been discovered. Additionally, based on the results of our collaborators, 16 freshwater operational taxonomic units were identified, which originate from water bodies of Antarctica, Chile, Norway, Svalbard, Canada, Japan, and France. The freshwater clones TL2 and TL4 discovered by us are the first morphologically identified representatives of telonemes inhabiting freshwaters. As well, we have investigated the ultrastructure of the found protists, and there are a number of unique features of ultracene structure, such as the intricate cytoskeleton and multi-layer corset. And Telonemia also has the similarities with other protists. For example, peripheral vacuoles, similar to cortical alveolus of alveolata, and tripod tubular hairs, similar to the Mystigonemus of Straminapilus, and extrusomas, similar to extrusive organelles of some Rhizaria, cryptomonads, and cataplepharids. Here. So, due to the sister position of telonemes in respect to SAR, these features of morphological similarity may be homologous, and this explains the origin of important synapomorphisms of SAR representatives from a common ancestor of the group, and thus, telonemia occupies an important position to examine the early evolution of SAR, which includes about 100,000 of one of the most relevant for the biosphere species. And in a more general way, our research emphasizes the importance of telonemia to investigate evolution of other important eukaryotic cells features, for example, their cytoskeleton, underlying their morphology, and cell motility. Considering a complex multi-layered cytoskeleton, we can assume that telonemia kept traits of organization of the ancestral cytoskeleton complex before its elements were differentially reduced in other eukaryotic groups. Further research by further research of cell anemia could provide a deeper understanding of evolution of the cytoskeleton and other cellular system of microbial eukaryotes. Thank you for attention, and if you have any question, I'll be glad to try to answer.
Yeah, no, fantastic presentation. It was very interesting.
Okay,
So what is the size? I know I was looking at these photos there. You said it was about 10 microns for some of these. How do you, when you do the genomics and stuff, you're just using one cell from a crude culture, or do you actually have these in laboratory cultivated in the, or how do you grow them even?
Thank you for your question. Yeah, we have culture in our laboratory, and when we find such interesting organism, we isolate it in the culture, and then we make molecular analysis using it. Yes.
Oh wow, huh? It's really neat. Any other questions or comments or thoughts? No, pretty good. All right, we'll move on to, here's another name I'm gonna butcher. I'm really sorry because you said your own name and it was not Anastasia. So I'm really sorry.
It's just short form.
I'm sorry about that. Okay, then Artem.
Artyom, please.
Artem, okay. Thank you very much for, yes, thank you for joining us.
Okay. Can you see my presentation?
No.
No.
No? One. What?
Looks like, yep, I can see it now.
Okay. Hello, everyone. My name is Artyom. I am a master's degree student of Penza State University from Penza City, and I work in laboratory microbiology of Papanian Institute for Biology of Inland Water. And now I would like to tell you about Vodonid's free-living heterotrophic flagellates. The Vodonids are a group of free-living heterotrophic flagellates which belong to class Kinetoplasteia, along with parasitic trypanosomatids. Currently, 3 monophyletic taxa of Vodonids are known: Eubodonida, Parabodonida, and Neobodonida. Sorry. Almost all representatives of this taxa are bacteriotrophic biflagellated protists, which play an important ecological role. It is known that Vodonids take up to 20% of the biomass of heterotrophic flagellates of aquatic ecosystem. Consuming bacteria, they contribute to the transfer of energy from a lower to a higher trophic level, being prey for predatory flagellates and ciliates, which in turn are eaten by small multicellular animals. Except for free-living representatives, kinetoplastids also include trypanosomatids, dangerous parasites of people and animals. Currently, according to phylogenetic data, the closest free-living relative of trypanosomatids is Bodosaltans, a bacteriotrophic biflagellated protist that together with Bodo uniculatus and several environmental sequences forms the clade Eubodonida. The lack of factual data on the morphology, biology, and ecology of the closest free-living relatives of trypanosomatids, in addition to the Bodosaltans, makes this question very relevant. Such studies can solve the problem of the origin of parasitism within kinetoplastids, as well as contribute to the stabilization of the phylogenetic tree of this group. Studying the ultrastructure of bodonids helps us understand the homology and origin of various cellular structures and forms a model of possible common ancestor of modern free-living and parasitic kinetoplastids. Besides, a more detailed ultrastructural study of bodonids can solve the problem of taxonomy of morphologically similar but genetically different species, for example, Neopoda designis. Among the distinctive features of bodonids, one can distinguish a well-marked cellular mouth, cytostome, continuing into the cytopharynx, mitochondrial outgrowth in the form of a kinetoplast, and lamellar crystal of mitochondria. The B75 clone we studied clone B75 was isolated and cultured for moist soil of Nepostayny Island in the city of Kiev, Ukraine, in 2014. For the cultivation of flagellates, the medium was enriched with Pseudomonas fluorescens bacteria. When working with clonal culture, we used light scanning and transmission electron microscopy methods. We also began to conduct a comparative analysis of the ultrastructure of this clone. Phylogenetic analysis based on ATS RNA gene sequences has shown that clone B75 forms a separate evolutionary lineage, is sister to the clade, which unites several environmental sequences and several species and genera of bodonids, including Neopoda barakensis, Neopoda curvifilus, Neopoda designis, Cryptolaxoides-like species, and genus Actuariola. The morphological data show a unique structure compared to the phylogenetically similar species of Bodonidae. The main distinctive feature here is a ventral furrow, this one. The general appearance of the cell is similar to the marine species Procryptobia sorokini and Neopoda curvifilus, as well as the freshwater Proboda caudatus. B75 differs from Procryptobia sorokini and Neopoda curvifilus by the larger cell size and the presence of a ventral groove and contractile vacuole. In contrast to Proboda caudatus, the anterior flagellum of B75 is almost permanently bent in a hook-like manner and never remains straight for a long time. Also, B75 cells do not group together or form cysts, as is the case in Proboda caudatus. All of the above suggests that clone B75 is probably a new genus and species within the family Neobodonidae. Thank you very much.
No, thank you. Those are really beautiful TEM photos. I had a question about kineto. I know you were just talking about bodonids and neobodonids, and the sister group, this kinetoplastids. These are very, I guess they're very famous terrestrial and possibly even freshwater parasites. Somebody a long time ago asked me about marine trypanosomes. Have you ever seen anything when you're looking through these things of like these bodonids, or what did the ancestor of that group look like? These terrestrial, was it a bodonid type group, or have you found anything that might be very closely related to kinetoplastids?
I think Dennis, help me.
Dennis.
Yeah, I need to translate probably. Тебе в морских водах трипаносомы где-нибудь попадались, вообще видел ли их, наблюдая за кинетопластидами, и как мог выглядеть их общий предок? Он больше на бодониду похож или на трипаносоматий?
I don't see in environmental sampling sample parasitic kinetoplastids, and I think that their ancestor
Mhm.
предок,
Ancestor, yeah.
their ancestor may be like Bodo Saltans, but him flagella, two flagella, but him second flagella как это исчез,
Disappeared.
disappeared, yes.
No, thank you very much. That's really, yeah, no, that was a long time ago. Somebody asked me if I ever see parasitic marine chenoplastids in what I study, because I study the invertebrates in the ocean. And some researcher asked me Julius Lukesh asked me he said, if you could ever find one, it would be very important. So keep an eye out for, he said, a biflagellate parasite in worms. And I was like, sounds good. I never found one. Anyway, no, thank you very much, Artem, for your talk. So next, oh no, I lost the list again. It is on the computer. Where is, oh, sorry. Well, next up we got Dimitri.
Yes. Hi.
So his screen is stuck.
Okay.
Oh, there we go. Okay.
I still cannot see.
Okay.
One second.
Okay, something is going on.
I saw it, but only for a second.
Okay.
Now it's.
My presentation is very hard. Okay, I try.
Yeah, now we see your screen, but it looks like it's stuck. Can you maybe start it once again?
Try.
Oh, there.
Or
Someone's running for help now. Is that what? Oh, it looks like it's. Oh, okay. I can see more of it now. Can everyone see it?
Yeah.
I can see it. Yeah, I can see it. Maybe try to click play, or I think it's looks good. Oh no, now we're back to no slides and no sound.
Yeah.
We've regressed.
Yeah.
Oh, there we go. Okay.
Okay. Turn on your microphone. We can't hear you.
Sorry.
Oh, there we go. Okay.
Dear colleagues, my name is Dmitry. I'm a PhD student from Voronezh State University. Now I live and work in Baroque as a junior researcher. I will present my studied on the diversity, morphology, and phylogeny of helizones. I study species diversity of centrohelid helizones, their distribution and different biotopes, and also study morphology of external skeletal elements of cell and phylogeny of centrohelids. Centrohelid helizones present a group of predatory free-living protists with radial symmetry. They float on a water column or roll over the substrate, capturing their prey with special structures, extrasomes. They feed on other eukaryotes, from small flagellates to multicellular organisms such as gastrotrichs and rotifers. Here you can see how the flagellate floating and touch the extrasome. The extrasome anchors the prey, and then the axopodium of the helizones is drawn into the cell, dragging the prey cells with it. Centrohelids don't chloroplasts, but they can often accumulate an intact plastids obtained from food in cytoplasm. The surface of most cells is covered with silica scales of various types. Organic spicules and mucopolysaccharide covers are also found. Phylogenetically, centrohelids are the closest relatives of haptophyte algae. Together with them, they form the Haptista group related to Ankyrocysts, Picazoa, and Tsar. Centrohelids are distributed all over the world. They live in fresh, brackish, marine, and hypersaline biotopes. They play an important role in the transformation and transfer of matter and energy in aquatic ecosystems. Along with diatoms, chrysophytes, testate amoeba, and other protists, they participate in the silica cycle. There is also a number of publications that show helizones as indicator organisms. An example, Raphidiophysis contractilis, that can indicate the contamination of water with heavy metal. At the moment, about 113 species of helizones have been described, but according to some estimate, we know only 10% of the total diversity. Despite the cosmopolitan distribution, the centrohelid remain poorly studied. For many regions, the data of this group are not available due to the small number of scientists research this group. Molecular study also don't cover more than 75% of the known species. In recent years, we have conducted a number of extensive studies on marine and freshwater centrohelids in Russia, Ukraine, Mongolia, Vietnam, South Korea, and some other countries. We found more than 75 species of centrohelids, and 11 of the study species were new to science. 32 isolated clonal cultures are cultivated in the collection of our laboratory. For some species that we know only from the place original description, we have added information about the morphology and expanded the knowledge of their habitat and distribution. An example from Rhinorhynchus scaposa and Pseudorhynchus formosa. This species was described from Chile, and then it were occurred in water bodies of Ukraine and Russia. Here you can, yes, Rhinorhynchus scaposa and Pseudorhynchus. Here you can see one of the species which was described by us as new to science. This is Pterocystis yunxuparki, isolated from South Korea. You can see a live cell, plate and radial scales, and the biotope in which was found. Besides the morphology approach, we also study phylogenetic relationships. At the moment, the small subunit ribosomal RNA gene was sequenced for 19 species for the first time, and phylogenetic trees were constructed. This allowed us to understand better the phylogeny within this group. New sequences allow us to stabilize the tree and show the presence of previously established clades with more statistical support. The existing of new clades that correspond to the rank of genera and families was also reviewed. I also plan to study in detail the features of the ultrastructure and of centrohelids and analyze the global distribution of the centrohelids according to metagenomic data. Thank you for attention.
Yeah, thank you very much. You're setting your what year of your PhD are you in it right now?
I'm a PhD student.
But what I guess how many years do you have in that program, or how many years do you have left? I guess is my question. Maybe dangerous question, but
4 years.
Maybe 4, yep. But like a few more years.
Осталось сколько? Осталось сколько?
1 year.
1 more year. Oh, okay. Are you going to be doing additional like multi-gene phylogenies, like the RNA sequencing on those heliozoans, or?
Дополнительно что-нибудь будешь, что-нибудь? Планируешь ли мультигенные исследования?
Yes. I have a plan also multi-gene sequencing.
Okay. So how in the heliozoans, they're not particularly well studied. There's some environmental sequence things, but not a whole lot of morphological data on them. What morphological feature is most important for understanding heliozoans? Is it the scales? Are those the most important taxonomic feature, or?
Scales of external skeletal have разные элементы скелета, то есть
different elements of skeleton.
Хорошо, как это? Я по-русски слово вспомнил. Sorry.
He forgot how to say it in Russian even.
I know it's hard to get questions about when you're going to graduate and what's the most taxonomic important question. But thank you for that.
Какие признаки важны для систематики?
Короче, form of radial and plate scales in external skeletal коррелирует, хорошо коррелирует.
It's correlate.
Correlation.
There's a correlation.
Correlate with molecular data.
Okay. All right. All right. Well, thank you very much for your presentation. The pictures were gorgeous. I really like the heliozoans. They photograph very well. And I think the last person we have is Olga, is going to be doing her presentation.
Hello.
Hello.
Dmitry, can you unlock your screen? Can you close your presentation?
Mitya.
Okay.
Hello, my name is Olga. I'm a PhD student from Voronezh State University. I work in microbiology laboratory, distribution, ecology, and phylogeny of the state amoeba. State amoeba are a polyphyletic group of protists. These organisms have a protective shell that they build themselves. The shells can be organic or agglutinated. Agglutinated shells are composite of self-secreted silica scales or mineral elements. The organism itself occupies only a part of the shell. The protoplast is attached to the shell walls using epipodia. These are only pseudopodia outside. Pseudopodia are of two types. These are lobopodia and philopodia. The state amoeba are polyphyletic. Most of phyllos, the state amoeba belong to the supergroup Rhizaria. The lobos, the state amoeba belong to the amoebozoa within Amorphea supergroup. The state amoeba have worldwide distribution and form diverse and abundant communities in freshwater and terrestrial habitats, including soils, mosses, stagnant water, and sewage water plants. The state amoeba feed on a wide range of organisms, including bacteria, fungi, algae, other protozoa, micrometazoa, and may consume rich organic matter. The total number of the state amoeba species can be estimated as 2,000. The state amoeba is the dominant protist and most important microbial consumer in peatland ecosystems. The state amoeba play important role in the cycling of elements in terrestrial ecosystems. They play an important role in aquatic and soil ecosystems as a key component of the microbial loop. The state amoeba can be used in applied studies as environmental and paleoenvironmental indicator and for forensic purposes, as well as for biomonitoring and ecotoxicology. I have even investigated the species composition, morphological features, and distribution of the state amoeba in the box system of the Osmansky pine forest, Europe and Russia, depending on the hydrological regime. I used the methods of light microscopy and electron microscopy. These are my photos of the shells of the state amoebas. I have studied, studied the species composition of 7 types of microlandscapes with different hydrological regime. Here you can see the structure of communities from the sphagnum, inhabiting the wet mosses, inhabiting the flooded area of the swamp, inhabiting decaying plant residue in water, inhabiting forest litter. As a result, the dominant, co-dominant, and rare species were revealed in each type of habitat. The abundance of all species of the state amoeba was obtained for each type of microlandscape. Obligate sphagnobionts and species of the state amoeba inhabited all types of microlandscapes have been identified, and the state amoeba with maximum and minimum density. Currently, I am studying the species composition and abundance of the state amoebas on 7 transects of the Azov Sea, coast from marine littoral to dry soils. The data from my research will be used in the complex project about the organization of food webs on the seashore and problems of transfer of matter and energy between terrestrial and marine ecosystems. Thank you.
So I had one question about testate amoeba. Your sampling area is around a single area, I guess. I don't know, how big is your sample area that you're collecting samples from?
Насколько велика площадь, с которой ты собираешь пробы?
Чем больше, тем лучше сейчас. Зависит от цели.
Yeah, this depends from goal of investigation.
Okay. Is there a in testate amoeba in different biomes? I guess the testate amoeba have very different species composition and diversity. So I guess you're if you're studying one area, do you think that would I guess negatively impact your study, or should you have a larger area? Because Russia has actually a very high latitude, they have a diversity of latitudes to get stuff from, either very high Arctic or even down lower.
Поняла?
Нет.
Раковины амебы населяют разные биотопы. Если ты будешь изучать маленькую какую-то площадь, ты найдешь мало видов, и нужно больше разнообразия биотопов изучать, или как?
Сейчас.
It's very interesting question,
Сейчас я по-русски сначала, потом не в обширности территории, а в виде биотопа. Чем больше видов биотопа будет, мох, лесная подстилка, почва, тем будет больше видов.
The main factor of the diversity of the amoeba is number of different microbiotopes, like in a small scales. The more microbiotopes we sample, more species we will find, and it's doesn't matter on like which territory in terms of like 1,000 kilometers.
Okay.
Just pick several microbiotopes within one book, and you will find a huge diversity.
Really? Okay, that's interesting. All right. Yeah, no, thank you for your
The last two speakers actually, they learned German at school, and they only started to learn English here during PhD, so that's why we have some troubles. but
No, you guys
getting
did.
better.
You guys actually did. There was absolutely no problem understanding your talks. You guys did phenomenal. So thank you very much. No, but anyways, I guess because it's a little after 6 p.m. here. And so tomorrow, I guess we'll see you guys at same time again, pending a power outage or natural disaster in Russia that prevents, or Japan
Yeah,
for that
I
matter.
hope.
So, and then the topic for tomorrow, I'll just be leading more or less a discussion about the goals for the students, goals for research, goals for the project. And then we can talk about how we can collaborate on this topic about the shellfish and bivalves in Japan. And then so we can just see, well, it'll more or less be a discussion about that I'll prepare and facilitate. So thank you.
Yeah, Kevin, should I prepare some presentation for tomorrow?
If if you could bring the any type of information or ideas you have about especially getting samples in Russia, how that might be possible. I know it's this with the coronavirus, it's very difficult, maybe impossible, but just hypothetically thinking about how to get the samples in Russia from the shellfish, from the aquaculture places. That would be something good to discuss. And then I'm thinking about how and when to get samples here in Japan and what's the most practical option for getting those, how to process it, and then once it's processed, how to deal with those samples effectively, being that borders are essentially probably going to be closed. I mean, through 2021. That's kind of what it's, I don't know, maybe I'm just pessimistic. Maybe everything will open back up in one fine day. But yeah, well, I guess just think about where to get samples and how to process it. Okay. And then how to exchange material and data or data would be that. All right. And then,
okay, great.
Yeah, thank you very much, guys. I really appreciate your talks. So yeah, the Hokkaido University students, you guys, everyone, thank you so much. You guys did really good. So I'll see you tomorrow.
Thanks. See you tomorrow. Bye.
All right.
Bye-bye.
Bye-bye.
Bye.
Bye.
Bye, guys.
Bye.